So now I think we need to have you stop sharing so that we can pass the baton to Ryan. He's our last speaker of the day. So Ryan is obviously not a dyed-in-the-wool DEL practitioner, but we brought him in because Ryan has recently moved from academia to Amgen, where he is running and heading up the Induced Proximity Platform. And Amgen has devoted a good amount of resource recently to acquiring new evolution and bringing in internally the DNA-Encoded Library technology to support their discovery efforts. So we're here to hear kind of the future of using this type of technology plus what exists at Amgen to move into new drug space. So Ryan, it's over to you.
Thank you, Gwen. Thanks for everyone for sticking around here. Hopefully the slides are coming through well for you. We're gonna change direction a little bit here. As you heard, I'm not a DEL expert at all, but we use DEL a lot in my group for executing on projects. I'm really here to talk about sort of the application side of what can you think about the DEL technology and how you apply it. And hopefully I'll excite you into what the future may hold here and get you excited about some things that you can do within your groups. So as I mentioned here, the title of the talk is Induced Proximity Medicines, a great fit for DEL technology. And you've already heard some of this concepts already today. So to get started here, I want to bring this slide to your attention because I think it's very useful to start with, and that is when we think about drugging therapeutic targets of interest, the reality is many of them are just classically undruggable by conventional medicines. And so here is sort of a graphical way of representing that, where typically we think of somewhere at most 15% of the current proteins that are druggable with conventional medicines. And that leaves, of course, a large portion of proteins that are really not druggable normally, but maybe we can have opportunities to capture those with novel approaches. And in particular, I'm going to be talking about this concept of induced proximity medicines. And so what are induced proximity medicines? Are really matchmakers, as this middle diagram is trying to elucidate. An induced proximity matchmaker medicine is really composed of two parts. One part that binds to the target of interest, where you want to have some kind of action. And so you see that highlighted here as the number 2 blue molecule. This is connected to another portion that is binding to an effector, typically a cellular effector in the cell. And by linking together sort of the 1 and 2 sort of warheads to form what I'm referring to here as an induced proximity medicine, you now bring in proximity the effector to the target, where the effector can now act on that target. And obviously, depending on what the nature of that effector is, you can have various outcomes on the target. And a few of these types of outcomes are highlighted over here. You've already heard today about the one of these, and that is the destruction of that target, removal of that target from the cell. And in this case, you can think of recruiting an effector such as ubiquitin ligase for modifying that target with a ubiquitin moiety. But you can imagine other scenarios of induced proximity. We have other types of effectors. And another example is, what about if you brought a kinase or phosphatase to this target to allow maybe activation or inactivation, depending on the nature of how you're trying to control this target. Or you can imagine other scenarios like you trying to relocalize that target. And these are all becoming potential realities, as there's a lot of development in this area. And I've just highlighted a few of these types of molecules, and you've heard already about, for example, the concept of around PROTACs. So, just to highlight a little further the potential here and why induced proximity is going to allow us to access sort of typically undruggable proteins. I've highlighted that here. I've already mentioned sort of typically you can imagine in the concept of sort of a classical protein drug interaction, you really require sort of a pocket in which a small molecule, in this case, binding actually has a functional effect. And so that, of course, is really limits the target space, if you will, of proteins that will likely have this kind of feature. And as I mentioned, many proteins sort of fall into this undruggable class because they just do not have such functional pocket. And so that's an opportunity for us to think about how do we capture some of these kind of proteins. In addition, I've highlighted 3 other examples on this slide of other contexts where using an induced proximity approach may allow you to overcome hurdles that are often seen in classical sort of conventional drug discovery. The first is this concept of compensatory upregulation. So imagine you could bound a molecule into this functional pocket and achieve the outcome in maybe an inhibitor, for example. But the cell responds by just shooting up the levels of that protein and thus making a stoichiometric type inhibitor very challenging. But if you can drive an induced proximity event like, say, protein degradation, you can just remove that protein from the cell as a whole. Or think of a context where you have the need to be very selective in targeting very similar proteins, maybe they're paralogs or proteins in the same family, but it's really hard to get that paralog selectivity just through binding of this one small molecule because the binding pockets are very similar. Now, if you now link this so that you require an effector molecule to come in, you can achieve sort of some of this selectivity on the back end, if you will, on how that effector interacts with the target differently between these 2 paralogs. Or finally, think about the concept where many targets, although they may be druggable, they're just not good targets because of safety and toxicity concerns. You heard a little bit around that concept in the last talk. But what about if you could target those kind of, if you could drug those kind of targets in very specific diseased tissue or cell populations, so that you could overcome this general toxicity issue and instead target it to the right place. And so again, the use of induced proximity where you rely on effector molecules, effector proteins that are highly specialized and specifically expressed in the relevant population is one way to allow you to engage these kind of targets. So we really believe that this concept of induced proximity is really the next wave of drug design and discovery. We believe that, as outlined in a recent review by Ray Deshays from Amgen, that induced proximity medicines may be the sort of 4th wave of drug discovery. We can define sort of the 3 first waves of drug discovery as starting, artificially here, sort of at the start of this graph as the inception of aspirin in the late 1800s, very early 1900s, was sort of where you had defined molecules, but you really didn't know what you were targeting. So you had defined molecules, but undefined targets. And this, of course, was the way everything worked for a long time, and it was sort of this empirical pharmacology approach. But then came the revolution of molecular biology and the biotech boom, where now here in green, you started in sort of the 80s, being able to now define the targets and of course, the defined molecules. And that took hold obviously very quickly. And then, that's obviously been very successful. And since then, more approaches have come around, such as biologics, which has been gaining since their initial in the early 2000s started to take hold. We believe that now that the sort of the 4th wave now is the concept of induced proximity in medicines that allow us to gain a greater traction over difficult drug targets. This is already being played out in the clinic, and here is just several approved drugs that work by the concept of induced proximity, and those approved drugs are indicated over here. I won't go through all of these examples, but they all work by sort of this target effector relationship. And I'll just point out one, the first one on the list, the thalidomide, which actually works by a protein degradation mechanism, where it essentially works as a glue molecule that sticks together an effector, a ubiquitin ligase, known as CRBN, to a target. Many transcription factors, in particular the IKZF family of transcription factors. But it's been clear that one thing that these mechanisms, these drugs, the mechanism of these drugs was only discovered sort of retrospectively and not necessarily in that prospective design. And that is something that is a big challenge for this field, and where the DEL technology can help with. So you heard about protein degradation as sort of I would say, the tip of the iceberg for induced proximity drugs. And you heard about PROTACs and glue-type molecules that stick together ligases and targets from Gwen. But I would say there's also a number of emerging targeted protein degradation strategies that leverage other machinery in the cell, in particular degradation through what you can think of as the cellular trash can, the lysosome, whereby you can drag proteins into the lysosome by a variety of different ways, either through endocytosis, in which proteins either on the surface or in the serum are coupled to membrane receptors like the M6PR receptor shown here that get internalized and dragged into the lysosome for ultimately degradation. So this is really an induced proximity event. The formation of autophagosomes around your target or your diseased organelle, for example, for degradation in the autophagosome lysosomal system, or even the recruitment of ligases to membrane proteins like here, the PD-L1 receptor, where it leads to the ubiquitination and then the ultimate endocytosis and degradation of PD-L1 in the lysosome. So this is becoming a really prominent way for controlling protein abundance in the cell in a therapeutic approach by using this concept of induced proximity. So I would say it's, as I mentioned, it's only really the tip of the iceberg when we start to think about this concept of just, pulling two things together. You can think of a lot of different ways for that to have a therapeutic outcome of interest. And I've just highlighted a couple ways here, such as protein stabilization, regulating protein activity, localization. I've highlighted some of these. And I think we should also be thinking about the target in these cases doesn't necessarily just have to be a protein. It actually could be an RNA. And where you can think about localizing different proteins to the RNA to achieve your desired outcome. And these are definitely not entirely theoretical. There's many examples now in the literature, and I've put several here to give you a flavor of how people are thinking about this concept of induced proximity. The first is just an additional play on the concept of protein degradation by PROTACs, but now in specifically linking small molecules to RNA to bring that ubiquitin ligase for ubiquitinating RNA binding proteins, which classically have been very difficult to drug, and that ubiquitin transfer obviously leads to degradation. But maybe you don't want to degrade your protein. Instead, you want to stabilize it. And this is a concept that has been recently put forward by a couple of folks, including Dan Nomura's group, where they showed that molecules they refer to as dub-tacs, which you have a ligand that binds to a target, in their case, for example, CFTR, is linked to a molecule here that binds to a deubiquitinating enzyme. And so that induced proximity of that deubiquitinating enzyme to the target can chop off those ubiquitin moieties and thus stabilize the target. And thus, of course, in the case of CFTR, increase the abundance of the protein, which is sort of the disease-causing state. You can think about other concepts where instead of you removing something, you're adding back. And in this case, it's this recent report of ACE-TACs, where you're driving the acetylation of proteins in a context and temporal specific way by recruitment of acetyltransferases like P300 CBP to targets of interest to induce this acetylation. You can drive the same kind of processes through the modification of proteins by phosphorylation. Here's the context of recruiting a kinase to a target to allow phosphorylation. This is the reverse, where you're recruiting a phosphatase to a protein to remove that phosphate. And you can do that sort of with both intracellular phosphatases, also with membrane phosphatases. Here's an example of CD45 phosphatase that has an intracellular phosphatase domain that has now been recruited to PD-1 to dephosphorylate it and therefore regulate the activity and stability. And finally, I'll come back to this point I made earlier that it doesn't just involve proteins, but you can actually target RNAs. And this is with a class of small molecules known as ribotax that has been popularized by Matt Disney's group at Scripps, Florida, in which you can think about an RNA that you might want to regulate. In this case, you might want to degrade it. And you can make a small molecule that binds specifically to that RNA that has another moiety sticking off of it that then binds to a nuclease and recruits that nuclease, and it chops this RNA up. So there's a lot of potential opportunities here, and I just would encourage folks to think about that you can come up with creative uses of induced proximity to allow one to tackle the difficult to drug targets, and it's really a new wave in this approach. And I think last two slides here is I want to point out that induced proximity extends beyond this sort of effector-target relationship. That in and of itself is very powerful, right? Because you're utilizing and harnessing the power of the cellular machinery to do the job. The molecule doesn't have to do all the heavy lifting. But I think you move beyond that to think about how induced proximity can be utilized. And I'm showing you two examples where you can essentially think about just gluing two things or a complex together. On the left is a really nice study coming out last year of small molecules that can drive the assembly of particular complexes of PP2A. This is a phosphatase in the cell that assembles into a trimer where there are various pieces put together, and there could be different flavors of that trimer. And what they came up with was small molecules that can drive the assembly of specifically this alpha form, and you can see it sort of highlighted up here. The second is the sort of opposite scenario. Instead of stabilizing this one form of the complex and therefore having more activity of that complex, you can think about the opposite, where you can glue a protein to something else to inhibit it. In this case, a well-known small molecule trametinib, an inhibitor of MEK1, actually turns out to function as a glue to stick MEK1 over here on the left in pink to a scaffolding protein KSR1, where the molecule interacts on both the MEK1 side. Here, this is the pink part of the molecule interacts with MEK1, and the blue part of the molecule interacts with KSR1, and this is still stuck in the middle. So you can think about how these glue-type molecules build on these existing but weak interactions between these complementary surfaces. And this can be used for a lot of different approaches. But for example, think about trying to correct mutations, where oftentimes these mutations, the missense mutations, may disrupt the function of the protein by preventing it from assembling with the right partners in the cell. But maybe now by sticking a small molecule there that sort of decorates off of the protein, just like a post-translational modification or like the more correct amino acid, you may now be able to reinstate these type of normal protein-protein interactions to achieve the undesired biological function of the protein. So this is my last slide, and I wanted to just touch again on a few concepts you've probably already been heard of and been thinking about, and that is, Dell technology is really enabling for induced proximity drug discovery. Obviously, these medicines are typically small molecules, so that fits very well into Dell. You already heard Gwen mention this, but I think this should be reiterated. We're really looking for ligands that do not need to be functional binders. I can't emphasize that enough, and that's where obviously DEL technology can be of high utility. It's already been pointed out that knowing where the DNA attachment provides an exit vector where you can start to assemble the two small molecules together in a way that'll be productive. And oftentimes the affinities of the ligand straight out of the DELs is sufficient to make tool molecules. And I think you should think about also the concept that you can use DEL screening for not only binary interactions like compound to protein, but also you can think about ternary complex screening where you might look for a compound that can glue together two proteins. And I think DELs are a great opportunity for this. So I'm probably over time here, but I'll stop and thank you all for listening and the invitation to talk, and be happy to take questions. Thank you.
Great, thanks, Ryan. I think that was a really good summary of many of the different things that we can do with a discovery mechanism that gives us binders to protein surfaces.